Welcome to The Explainer. Today we are going deep. We're journeying inside the microscopic world of the cell to figure out how a single set of instructions can build, well, everything from a simple bacterium all the way to you.
So, what's the connection? I mean, really? Well, the answer is that you are all made of cells. These are life's most basic building blocks. But here's the thing, not all cells are created equal. They come in this incredible variety of shapes and sizes, and each one has a super specialized job. So let's start by learning how to tell them apart.
All right, our first step is to start thinking like a biologist. We're going to focus on the key features that divide the entire living world into its biggest, most fundamental categories. You know, biologists make this look easy, and they do it by focusing on just a few key questions. Looking at this chart, we can actually classify these different specimens just by answering yes or no to a few things. But I want you to pay really close attention to that last column, DNA and nucleus. That right there, that's the single most important distinction in all of this. Yeah, that one little feature creates the biggest dividing line in biology. It's kind of the difference between having your master blueprints just scattered all over the factory floor versus keeping them safe and sound in a central office. So if you look back at our chart, cell 3, the one that's missing that nucleus, that's a perfect example of a prokaryote. And even within the eukaryotes, the cells that do have a nucleus, there is amazing diversity. A plant cell, for example, is like a little fortress. It's got a rigid cell wall for structure and these things called chloroplasts that act like tiny solar panels. Animal cells, like the one you see on the right, are way more flexible and they just don't have those features. Okay, so we've got our cell, our little city, but how does it interact with the world around it? How does it get supplies in and ship its products out? Well, it all happens at the border, the cell membrane. Think of the cell membrane as a really sophisticated bouncer at a club. It is not a solid wall, not at all. It's this selective barrier. It lets some molecules pass right on through, no problem, but it stops others completely. And it even has these special doors just for very specific cargo. So let's look at the two main ways this whole thing works. This is the easy way in. It's called passive transport. Particles will naturally just spread out from where they're really crowded to where there's more space. It's a process that requires zero energy from the cell. It just happens. And this is how small molecules like oxygen can just drift right across the membrane. But what if the cell needs to stockpile something? What if it needs to pull in a resource from an area where there isn't very much of it? Well, that's like trying to push a boulder uphill. It's not going to happen on its own. The cell actually has to fire up a special pump and spend energy in the form of this molecule called ATP to literally force the particles across the membrane. Now, this whole process brings up a really important special case of passive transport called osmosis. Water is always trying to balance things out. So here, you can see the solution outside the cell is much saltier. So what does the water do? It rushes out of the cell, causing it to shrivel up. This is the exact same principle behind preserving food and salt, and it's absolutely essential to how our kidneys function. All right, now we're going to go from the city's border, the membrane, straight to City Hall, the nucleus. This is where the master blueprints are kept. We are talking about DNA, the instruction manual for life itself. This right here is the fundamental workflow of life. The DNA holds the permanent master plans. To actually build something, a temporary copy of one of those plans is made. That's RNA. And then that copy is taken to the factory floor to build the final product, a protein. So the very first step is called transcription. Inside the safety of the nucleus, a specific gene on the DNA gets unwound, and a little messenger molecule called mRNA is built to match it. You can think of this mRNA as basically a disposable photocopy of a single recipe from a giant, priceless cookbook. That mRNA messenger then leaves the nucleus and it travels out to a ribosome, which acts as the cell's protein factory. And here, the recipe is read, 3 letters at a time. For each 3-letter word, a specific building block, an amino acid, is added to this growing chain. And that process is called translation. But what happens if there's a typo in the master blueprint? I mean, a mistake in the DNA sequence. These mistakes are called mutations, and they can be totally harmless, or they can have really dramatic effects on the final protein. To understand mutations, we need this. It's basically a decoder ring for the genetic code. By finding the first letter of an mRNA sequence on the left, the second letter on top, and the third one on the right, we can translate any gene. This thing
actually lets us predict the exact outcome of a typo in the DNA. Okay, let's trace this like a case study. The original DNA sequence is TTC. But in mutation 1, a single letter changes. That middle T becomes an A, so the new sequence is ATC. Now, let's see what happens when the cell tries to read this new code. That single letter change is absolutely critical. When the cell reads the mutated DNA, ATC, it creates an mRNA message of UAG. And if we look that up on our decoder chart, you can see it doesn't code for another building block at all. It codes for stop. This one tiny typo tells the factory to just shut down production, which results in a shortened and probably useless protein. Okay, finally, let's zoom all the way out and see how all these components we've been talking about, the membranes controlling transport, the DNA holding the code, and the proteins doing all the work, how they all come together to create a living, functioning cell. And this shows us a universal rule of life. Those proteins we just learned how to build, especially the workhorses we call enzymes, are incredibly picky. The enzyme for photosynthesis in this plant works best at one specific temperature, while the pepsin in our own stomach needs a super acidic environment. You change the conditions just a little bit, and the cell's machinery just grinds to a halt. So, to grow and to repair tissues, our cells have to make perfect copies of themselves. This process is called mitosis. And it's this elegant cellular dance where the cell first duplicates all of its DNA, then meticulously lines up all the copies right in the middle, pulls them apart to opposite sides, and finally divides into two brand new identical cells. And that brings us to a really fascinating point. Think about it. A muscle cell in your arm and a liver cell in your body have completely different jobs, they look totally different, and yet they contain the exact same DNA blueprint. The complete instruction manual is present in every single room of the house. And that leaves us with one of the most profound questions in all of biology. If every single cell has the same master cookbook, what is it that tells one cell to only read the nerve chapter while another one only reads the skin chapter? The answer lies not just in the code itself, but in how it's read. And that's a puzzle for our next explainer. Thanks for joining us.